In this video, I'll discuss the classification of amino acids, which are the building blocks used to construct proteins. Why do we bother classifying amino acids? It might be helpful to consider an analogy. Let's say different fields of nursing. While two psychiatric nurses have their own distinct manner and personality, their skill set and the care they provide to their patients will likely be more similar to each other than to nurses who work in completely different fields, say acute care or pediatric nursing. Likewise, of the 20 different amino acids commonly found in our proteins, some share features that enable them to perform similar roles within a protein. We will classify the amino acids into three major groups: the polar charged amino acids, the polar uncharged amino acids, and the nonpolar amino acids. Things that are nonpolar are hydrophobic or water-hating, while things that are polar are hydrophilic or water-loving. Here, I've represented amino acids with circles, with the hydrophilic amino acids shown in blue, and these three, the ones in peach, are hydrophobic. For this chain of amino acids to become a functional protein, it needs to fold up. Keep in mind our proteins are most often in a watery environment or aqueous environment, like the cytoplasm of a cell. The hydrophobic nonpolar amino acids prefer to get away from water and congregate at the protein's core, while the hydrophilic amino acids can sit at the surface of the protein in contact with water. Before classifying amino acids into these three groups, let's look at the features that all amino acids share. Amino acids have an amino group that contains nitrogen. That's where the amino in the name comes from. What's the acid component? All amino acids have a carboxyl group. This carboxyl group tends to give up a proton, which is the defining action performed by an acid. That's where the acid in the name comes from. In fact, at physiological pH, the carboxyl group is most often negatively charged because it tends to shed a proton. The amino group behaves in an opposite manner and tends to pick up a proton at physiological pH. This gives it a positive charge. You may see amino acids represented with or without these charges indicated, and both are correct. The carbon in the middle is referred to as the alpha carbon, and all amino acids also have a hydrogen attached to their alpha carbon. Together, the components shared by all amino acids make up the backbone of a protein. The alpha carbon can form 4 covalent bonds, and this fourth spot is where the 20 amino acids differ from one another. We have represented this variable group with R. The variable group is also sometimes referred to as the side chain. To examine the features that make an amino acid nonpolar, polar with a charge, or polar and uncharged, it is necessary to focus on the R group. If I focus only on the R group and see that it consists only of carbon and hydrogen, it is nonpolar. Carbon and hydrogen share electrons pretty equally, so this portion of the amino acids will not dissolve well in water. The R groups of amino acids that are nonpolar can form hydrophobic interactions that stabilize tertiary structure. If the R group has oxygen or nitrogen in addition to carbon and hydrogen, it is polar and hydrophilic. Oxygen and nitrogen are more electronegative than hydrogen, which means that they tug on electrons more strongly. In some cases, this can result in a full charge, which is indicated with a positive or negative sign. Here, we see a negative charge. The R group of charged amino acids can form ionic bonds that stabilize tertiary structure. Sometimes, the unequal sharing of electrons doesn't result in a full-fledged charge, but still imparts what we can think of as partial charges on a molecule. In this case, the oxygen is partially negative and the hydrogen partially positive. If you encounter an R group that has oxygen or nitrogen and it does not have a positive or negative sign, it is polar and uncharged. Polar, uncharged amino acids can form hydrogen bonds to stabilize tertiary structure. I hope that this video has been helpful, and thank you so much for watching.